Welcome. In this session, I'm going to discuss about two major groups of kingdom Monera. So let's see. This kingdom Monera includes two major groups of bacteria. One called Archaeobacteria and another one Eubacteria. So here the Archaea from Greek, which means ancient, whereas here from the Greek eu, which means true. So these are ancient bacteria and those are true bacteria. So here coming to the archaeobacteria, so these bacteria live in extreme environmental conditions. So those extreme environmental conditions, I call them harsh environments. Harsh environments. So harsh environments, like example, in hot temperatures where the temperatures goes beyond 100 degrees Celsius, or the temperatures in minus in snow. Okay, like this, these conditions, even in deserts where the temperatures will be very hot. Hot springs are the locations where we see temperatures going beyond 100 degrees Celsius. So here harsh environments, like example hot springs. So in this case the temperatures goes beyond 100 degrees Celsius. Okay, in deserts, in snow, so etc. are harsh environments. So these bacteria, the bacteria belonging to the group Archaeobacteria, they live in such harsh environmental conditions. So how they are able to live and what makes them to live in such harsh environmental conditions is due to presence of a different cell wall. So these bacteria have a cell wall with a different structure. So due to having a different structure of the cell wall, these bacteria are capable of living in such harsh environmental conditions. Usually the bacteria cell wall will have a substance called peptidoglycan. So peptidoglycan is present in cell walls of bacteria, but the archaeobacteria don't have that peptidoglycan. So here, as I mentioned, they have a different, okay, cell wall, okay, structure wall I'll mention, instead of a different with a different composition. So with a different composition, okay, so that is, they have polysaccharides plus proteins. So they have polysaccharides and proteins without peptidoglycan. So without peptidoglycan. Whereas the other bacteria, the eubacteria, they will have peptidoglycan in their cell wall, but the archaeobacteria will not have, right, the peptidoglycan. So instead they'll have some polysaccharides and proteins, and this arrangement of substances in the cell wall makes these bacteria to live in such harsh environmental conditions, right? Now these archaeobacteria are classified into three different categories depending on the habitat. Depending on the habitat, So they are classified into three categories. So here habitat means location where the organism lives. So it's a place where the organism lives is what we call habitat. Habit is different, habitat is different. So depending on the habitat, the archaeobacteria are grouped into three different categories. One called halophiles and another one dermoacidophiles and then methanogens. Right. So here halophiles, which means salt loving. So this tells us that these are the bacteria that are capable of living in extreme salty areas. And coming to dermoacidophiles, so these bacteria, they like temperature and acidic loving bacteria. And coming to methanogens, these are the bacteria that produce methane. So these are methane producing bacteria. So methane producing bacteria are called methanogens. Okay, right. So the halophiles are the bacteria that live in extreme salty conditions. They live in extreme salty conditions. So those bacteria that live in extreme salty conditions, like for example Dead Sea, so Dead Sea has a more concentration of salt, so you may see bacteria present in those waters also, and you need to call them as halophiles because they like to live in that extreme salty conditions. So they are halophiles. Like example, in this case, halococcus. So halococcus is an example of this right halophiles. Even halobacterium also you can consider, etc. are some examples of these halophiles. Coming to dermoestophiles, so these bacteria that live in hot springs. So bacteria that live in hot springs. So hot springs are the locations near a volcanic eruptions where water gets collected and the water will be in a boiling state. So even so, it means that it has temperatures of 100 degrees Celsius, and even in that temperatures you will see some bacteria present in it. So those bacteria that live in such hot springs are called dermoacidophiles. Okay, so they live in hot springs, and here that's what they grow in. Okay, high temperatures. They grow in high temperatures and low pHs. They grow in high temperatures and low pHs. So here low pH is high acidic conditions. So which means acidic conditions. Right. So some examples of these dermoacidophiles like dermoplasma. So I'll just mention here example dermoplasma is an example. Okay. So this one lives in hot acidic springs. So it lives in hot acidic springs. Lives in hot acidic springs. Right. Then coming to methanogens. So these methanogens are bacteria that live in marshy areas. Bacteria that live in marshy areas. So a marshy area is a wetland. So wetlands are usually found, okay, near estuaries or near a river where the water flow doesn't take place. So as a result, the water will be stagnant all the time, right? And we will see in case of marshy areas, this location will be dominated by grasses. So you'll see grasses. Similar, you will see in case of swamps. Of course, they will have trees, but these marshy areas are mostly dominated by grasses. So you'll see, okay, so this is water, and these are grasses which are present. All the time, the water will be present. So this is a wetland. So a marshy area is a wetland. Okay. uh, and in these areas you will see the methanogens present, and where they will be producing the methane gas. So it's a marshy areas, right? So if you still find difficulty in understanding it, so just type in Google. It will show you the image of these marshy areas, okay, how they will be. So the bacteria that live in marshy areas, they are the methanogens here. And these methanogens are usually sensitive to oxygen. So they are sensitive to oxygen. Okay. And then they also don't have the peptidoglycan in their cell walls. I think I already mentioned that point. The archaebacteria are different from other bacteria. So in not having that peptidoglycan layer, then the methanogens, not only they are present in marshy areas, they are also seen present in alimentary canal of ruminant animals. So the methanogens also present in alimentary canal of ruminant animals. Present in alimentary canal of ruminant animals. So here ruminant animals are animals that chew cud. So like for example, you might have seen cows or buffaloes that they take up the grass, okay, and keep the grass in their stomachs, and then right, they soak the grass in the water, and then later sit in a shadow place, and then bring back the food into the mouth and start chewing. So that's called cud. Okay, so the organisms that chew cud, they are called ruminant animals. So in ruminant animals also this bacteria is present, so where they will be producing the methane gas. So here I'll just mention how they are going to produce that methane gas. So they are going to use carbon dioxide. So with the hydrogen they produce methane, okay, and some water is released as a byproduct. So this is a equation for methane, so production. So methane is produced, okay. So just for understanding purpose, I have given this equation, so to tell you that how the methanogens will produce this methane, right? Now examples of these methanogens: methanobacterium. Methanobacterium. Later in our later chapters, we will see that how we are going to use this methanobacterium in production of biogas. Okay, so that's about the three groups: the halophiles, the thermoacidophiles, and the methanogens. Then let's see the next group, that is eubacteria. So the eubacteria are true bacteria, and they have peptidoglycan in their cell walls. Okay, so that is, they have rigid cell wall. So they have rigid cell wall, and if they are motile, they will have flagellum. So if motile means movable, they have flagellum. So flagellum is a whip-like structure that helps in locomotion of bacterium. So it's a whip-like structure that helps in locomotion, right? Motile, which means movable. So if of course all bacteria are not movable, if those bacteria are movable, then they may have flagellum. I hope okay, in earlier sessions you might have seen a bacterium. So here is a rod shaped bacterium, and here this is the flagellum, is flagellum. Okay, so this is rod shaped bacterium. Okay, so the flagellum will help in locomotion, right? And these eubacteria are classified into two categories based on the nutrition. The eubacteria are classified into two categories based on their nutrition. So that is autotrophic bacteria and heterotrophic bacteria. So here already we have understood what actually is this autotrophic bacteria. So autotrophic bacteria are the bacteria that synthesize their own food materials. So here they synthesize their own food materials, right? Whereas heterotrophic bacteria, they depend on other bacteria for food. So they are called heterotrophic bacteria, right? So autotrophic bacteria and heterotrophic bacteria. So you can say these heterotrophic bacteria will not perform photosynthesis. So here no photosynthesis or even chemosynthesis. So whereas under autotrophic bacteria we have two categories, uh, the photoautotropic or chemoautotropic. So that is you can take as photoautotrophs and chemoautotrophs. So these autotrophic bacteria are of two types: photoautotrophs and chemoautotrophs. So are two categories under this autotrophic bacteria. So you can also call photosynthetic autotrophic bacteria. Photosynthetic autotrophic bacteria. In the similar way, you can also take these as chemosynthetic autotrophic bacteria. Okay, so anyway, I think it's not being covered, so I'll just remove it. So photosynthetic autotrophic and chemosynthetic autotrophic bacteria. So now let's see under this U bacteria, first photosynthetic autotrophic bacteria. Photosynthetic autotrophic bacteria, right? So here, this photosynthetic autotrophic bacteria, it includes cyanobacteria. So these cyanobacteria are commonly called blue-green algae. So in short, it's BGA. That stands for blue-green algae. So it includes the cyanobacteria here. Okay, and these cyanobacteria have chlorophyll A. So this chlorophyll A is a photosynthetic pigment. So it means that this is a pigment that is involved in the process of photosynthesis, especially in absorbing light energy from the sun. Okay, so that is chlorophyll A. Now the question comes that in other bacteria, whether they have this chlorophyll A. No, only cyanobacteria have this chlorophyll A. So chlorophyll A is a pigment that is also present in higher group of plants like in algae, bryophytes, pterophytes, and up to gymnosperms and angiosperms. They have this pigment, photosynthetic pigment chlorophyll A. While in other bacteria, they have bacteriochlorophyll. So this bacteriochlorophyll is a different chlorophyll pigment. So it is not like this chlorophyll, the structure is different. I will write in other bacteria. So they have bacteriochlorophyll. Right. So this is a photosynthetic pigment. The next one, these cyanobacteria are unicellular. They may be unicellular, means made up of only one cell. Their body is made up of only one cell, or they may be colonial. Right. So colonial, which means living in colony, or they may be filamentous. So filamentous, which will be like a chain. So when we see, so here the cells will be attached one to one, and it may result in a chain like. So this is a filamentous. And colonial form, where we see many living at one place, so this is a colonial form. And unicellular, which means having only one cell. So these are either unicellular or colonial or filamentous forms. Then these are found in fresh waters or in marine waters. So they are found in fresh water. So fresh waters will include like rivers, ponds, lakes. So these the waters present in that rivers, ponds, lakes, etc. is fresh water. So having a very less salt. So they are fresh waters, and also in marine waters. So it means that seen even in seas and oceans, we find these cyanobacteria growing. And we also see some of these cyanobacteria living even on land. So they are terrestrial. So they are also seen growing on land. So that we call terrestrially. So that is terrestrial blue-green algae. So we understand that they are seen in fresh waters, they are also seen in marine waters, and they are also seen growing on land. Then the colonies are usually surrounded by a gelatinous sheath. So the colonies or the filamentous forms are covered by a gelatinous sheet. So we will see example in this case, right, by taking nostoc. Covered by gelatinous sheet. And see here. So here nostoc is actually a filamentous form. So nostoc is a filamentous form, and can see, and this is covered by a gelatinous sheet, which actually helps in not getting dry quickly, so it retains water. So you can see here, this is a mucilaginous sheet. So we can take it as a, right, a mucilaginous sheet, and these are cells like we see in case of nostoc. Okay, then so here the some cells in the nostoc are able to fix atmospheric nitrogen. So those cells that are capable of fixing atmospheric nitrogen are called heterocysts. So you can see in this case, okay, so this is a cell that's capable of fixing atmospheric nitrogen, and that is called heterocyst. So heterocyst fixes atmospheric nitrogen. So what is this fixing atmospheric nitrogen? Is conversion of this, okay, nitrogen into ammonia, and this will be done in the presence of an enzyme called nitrogenase. So that nitrogenase is present in this heterocyst, and it will help in fixing this nitrogen. So nitrogen cannot be taken by many organisms like plants and animals directly. Only some microbes have that ability to convert this atmospheric nitrogen into ammonia, and then later occurs in form of nitrates and nitrites, which will be taken up by the plants, and from the plants, by eating on them, the animals will get nitrogen. So nitrogen is an important component in many biomolecules, like in case of DNA. So that's, I have given an example. Later in the chapter biomolecules, we'll see a lot many other examples. Okay, so here the nitrogen converting into ammonia is what we call fixing nitrogen. So fixation or nitrogen fixation. So this is called nitrogen fixation. It's called so nitrogen fixation, right? Then here these heterosis we can see, which I mentioned here in case of nostoc, and another example also I will give you, like in case of anabina. So heterosis are also seen in another bacterium called anabina. So in that also we find these heterosis here. Okay, right. So these are some characters of cyanobacteria, right? Now here some other examples of these cyanobacteria also we will see, like oscillatoria. I'll mention other examples. Here we have seen two examples, nostoc and anabina. So, and another example is oscillatoria, and then spirulina is another cyanobacteria. So this spirulina is edible. So humans will eat this spirulina. So it's an edible cyanobacteria. So that's about autotrophic, I mean photosynthetic autotrophic bacteria. Right, so with this, I will end today's session. In the next session, we will see about the chemosynthetic autotrophic bacteria.